I gave to this lesson is, does our brain control the immune system or vice versa? What we know, or you should know, is that immune system impairments accumulate with time. Recently, people talk about a contribution of immune malfunctioning to a lot of neurodegenerative diseases. So that raises the question, who controls it? Who knows who that is? Molière. Molière said all diseases start in our brain. And he proved it. He was a very well-known hypochondriac. And eventually he fell dead on the stage when he was playing in one of his plays. So even if you're a hypochondriac, you may die from whatever. And I need to confess that I started this survey and study from a very well-assured understanding that the brain controls the immune system. Today, I'm not that sure anymore. I'm convinced that there is communication and that perhaps sometimes the immune system affects the brain and contributes actively to neurodegenerative processes. So who controls it is still a question. We can discuss it at the end. So we're talking about functions that we do not think about. The brain controls not only those decision-making processes that we are aware of, but also functions that always need to be there, like breathing, like heart functioning, like the digestive system, like all of that is called the parasympathetic system. So we have two systems that balance each other, the parasympathetic and the sympathetic. Sympathetic is what you would call adrenaline, okay? And the parasympathetic is a balance. What does it control? First, it affects the brain. Both systems are managed in the brain as a center of command. So the brain sends a nerve called the vagus nerve from here all the way down to the digestive system. This controls, for example, salivary function. You watch someone eating and suddenly your mouth starts watering. You didn't think about that. You did not think, wow, I would like to eat this and then I would need to produce saliva. It just happens. You get into sunlight and your pupils constrict. Again, you don't think about that, it just happens. Digestive activity should be activated. If you watch a movie of someone eating, you'll get hungry. Your digestive system starts working. You didn't really mean to do that. We call this parasympathetic pathway the rest and digest pathway, whereas the sympathetic is what we call fight or flight. And the entire concept is that we inherited both systems from our ancestors, and reaction to environmental stimuli often meant in our ancestors' days that they need to run for their life. So if you have more adrenaline, your heart works faster, you run, you can survive. When that gets over, you need to quiet things, rest, and digest, okay? So the parasympathetic system is controlled by the neurotransmitter acetylcholine. So just in case you like chemical formulas, this is the chemical formula. Okay. So rest and digest, fight or flight, these are stress idioms. That's what we refer to when we talk about stress. What is stress? we copied the term stress from physicists. The physicists would tell you that if you apply pressure on a body, above a certain threshold, that body would break. This is applying stress. We adopted that statement for a psychological reaction. Someone applies psychological stress on a person, that person may also break, although metaphorically, but again, it's the same statement. So an individual might be aroused by an aversive situation. What does stress do? So here is the brain, and stress activates in the brain a pathway starting from this region in the brain, the hypothalamus over here, going into a gland which is here, the pituitary gland that produces hormones, and all the way to the adrenal gland next to the kidney. So that means that stress is not staying in the brain. It goes out of the brain and it comes back to it. This leads to alertness, which again we said we need to survive. It increases heart rate and blood pressure. It suppresses metabolic activity and immune system activity and reproductive system. You don't really think about sex if you have a lion running after you. So that makes sense. And people talk about an association between stress responses and neurodegenerative disease, which is why we are discussing it today. And we'll focus mainly on the suppression of the immune system as a mechanism of action that controls the consequences of stressful insults. And if you want to read more about that, we just came out with a book, and it's on sale at Amazon. So that's the start. What do we know about stress? This guy over here is Robert Sapolsky. He works in Stanford. He's a professor of psychology. He writes popular books about stress responses, and he writes in the New Yorker and in the New York Times. He's a super brilliant lecturer. Last time I visited him, he said, stress is an evolutionary reaction of survival. He said, if I would live in Germany in the 30s, I wouldn't stay there 1 week because I tend to get stressed easily. And those who reacted that way survived. So he believes that stress responses are still playing the ancient role of protecting the individual. And he says there are changes in hormone secretion and physiological consequences. And if you look at those, the consequences we just discussed, increased cardiovascular tone, initially immune activation, only in the long term you get suppression. Energy mobilization, loss of appetite, enhanced memory consolidation. This is worth discussing. Does any of you remember what you did the night that Rabin got killed? Or you were, okay. You remember what you did the night before? Of course not. This is what we call consolidation. So if something really traumatic happens, we'll remember not only that thing, but things that happened around it and are totally unimportant. In that night, the television showed Crocodile Dundee, which is a rather stupid movie. I would never remember which movie was shown on television. But the contrast was so drastic that this remained with me. That is what we call consolidation of memory. And loss of sexual behavior, we did say. What about, can we group these together? We will say that the physiological consequences of an acute stressful condition induces anhedonic behavior, the non-pleasurable behavior patterns. It would induce intrusive memories. So again, we'll remember things that happened around that event, and it would increase anxiety. And they would say that anyone who was subject to a drastic traumatic event would tend to be more anxious from that day on, which probably applies to many, many people in this country. So this is, again, something to remember. At the same time, there are changes in hormone secretion, which make all this happen. And there are many different hormones. You don't really need to remember the names. And most of them have activity on the immune system. So the connection between stressful responses and the immune system is very well established. Now, what about acetylcholine? Acetylcholine is secreted from the same vagus nerve that comes all the way from the brain throughout the body. And then it gets degraded. And a major way to control the signaling through acetylcholine is to change the level of the proteins that degrade it. These are called cholinesterases, okay? There are two cholinesterase genes. One is located on chromosome number 3, and there are a lot of mutations in it. Of those, one is really abundant in the Israeli population, like 20% of Israelis would have a less well-functioning cholinesterase, and 1 to 1,000 will have a real mutation with very drastically reduced function. And then there is another gene on chromosome 7, so another site in the genome. Again, there is a mutation. The mutations in these genes are more abundant in the Israeli population than if you compare to Caucasian non-Jews. Why is that? One thought is that it helped survive with the plants in the Middle East, solanum vegetables like eggplants, like tomatoes, which were not endemic to the region, but eggplants were, produce an inhibitor of cholinesterases. So if you're less sensitive to that, that may help. But inhibitors of cholinesterases are also insecticides. So what happens to people who carry such mutations and live in an area with a lot of insecticides being sprayed? What happens to them is, this is the electroencephalography. This is the insecticide they're exposed to. And what happens to them is they get hyperactivation of the frontal lobe over here that controls behavior, emotional reactions. They show down-regulation of deep nuclei, which control cognitive function and thought. And they do show impairments in memory and learning. So if you carry mutations in anything to do with environmental exposure, you would do better to live in Jerusalem, for example, where there is no agriculture. Okay, years ago, we asked the question, what happens in the brain under acute stress? And what you see here is the activity of the esterase, the enzyme that we are interested in the brain of mice that were exposed to confined swim. So mice can swim. They all know to swim. They don't like it. If you throw a mouse for 4 minutes to a water tub, they will swim and they'll be very upset. If you take them out for 4 minutes, warm them up, let them dry, and then do this again, this is really the end. And they get really stressed. And then what they do is they overproduce acetylcholinesterase. Because what happens when we are stressed? We need to react more immediately, more functionally to the incoming stimuli. So the brain produces this neurotransmitter, acetylcholine, a lot of it. But then something in the brain acts to balance how do you balance an excess of a neurotransmitter? You produce the protein that degrades it, okay? So here it is, over 2 hours, and then it stays rather high for a while, which means that after a stressful event, we tend to be depressed. And if you have a recollection of when were you stressed really acutely, you may remember that after that you didn't feel that happy. So, okay, this is what happens to the enzyme. What does that mean to the immune system? Now I need to tell you that in the circulation, we have white blood cells that act to protect us from infection and immune insults. What happened? Okay. 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 So we have here the brain. The brain sends through the vagus over here the neurotransmitter acetylcholine, and it sends it throughout the circulation. In our blood system, we have white blood cells. The white blood cells sit there and serve as sensors. Something is wrong, you got infected, you got wounded, the white blood cells produce immune proteins to protect from infection or from a wound. And this, remember, we started with the statement that stress reaction initially came to protect the individual and ensure survival under threat of wound. So a stressful situation, psychological stress, induces massive production of immune proteins. Acetylcholine blocks that because we don't need to waste energy on immune system. Now the brain takes control. So then acetylcholinesterase comes up. So the first thing that happens is the nerve cells throw out a small chemical, acetylcholine. The next thing that happens, the brain throws out a protein that breaks down that acetylcholine so that to quiet things back. And then there is nothing to block the immune system. So what we say here is there is a sequential process where the reaction to stress changes immune function in a process way. Okay. And then we thought, okay, if that's the case, then we can measure the enzyme activity in the blood. That biochemists know to do that. And then look at that as a marker of stress and inflammation or immune activity. And another thing we said is we would like to prove that. You know, a biologist today has a theory, a working hypothesis, and how do you prove that that working hypothesis is correct? What we do is we engineer a mouse, and we change the amounts of the protein we think is in charge of something, and then we say, if we produce more of this protein or less of this protein, would we change that function? So we said, okay, the brain produces a lot of this protein under stress, but is it really involved in creating the stressful reaction? So we engineered a mouse, and a mouse has an excess of the protein from the human system. Here's the mouse. And this is not super speed. This is real, the real speed of the animals. Did any of you ever see a mouse in a cage? Okay, so if you would see a mouse in a cage, they scratch a little, they think, they go to eat. They don't run like crazy. These mice run. They are 15% lighter than the litter mates that are not engineered. So they really spend a lot of energy. In other words, they behave as if they're anxious. How does it affect their cognitive function? So we put them in a maze. This is a maze. We put the mouse here, and we put some food over there, and the mouse needs to learn that if he runs all the way to the other end, he'll get some food, okay? So here is the mouse. You see the mouse? It runs, it learns the maze very well, gets some food, then we put it again. We do this 5 times. This is the normal mouse. Now look at the engineered mouse. See the mouse? It goes back and forth, and back and forth. It doesn't remember where the keys are, it forgot its glasses, it's stressed. So you would tell me, but that's not a big surprise. We too make errors of judgment and forget important things when we are stressed. True, but we didn't stress this mouse. All we did was to add this protein. So this is what biologists call a gain of function experiment. If this gene is important, then it would create the phenotype, the pattern of behavior, which is what it did. So the question now is, what happens to the immune system? So this is Adi, that's her PhD work. And she measured immune proteins, 3 different ones, in the blood of normal mice and in the engineered mice. And I think you can see for yourselves. These mice, their immune system works over time. They overproduce all of these proteins. Okay, so we say we have a beginning of evidence, what we call a proof of concept, saying that this system, the parasympathetic system, is associated with both psychological stress responses and immune reactions. Does that refer also to what we call post-traumatic stress disorder, which is the extreme manifestation of stressful situations? So, you know, I like to tell you a little bit about the history. So this was Walter Cannon, and he was one of the very first investigators to study stress responses in a thematic way. But if we look into the history, this is a picture from Herodotus. And Herodotus was telling about this soldier, Epizelus, who was behaving himself as a brave man should in a fight, in a battle, when suddenly he was stricken with blindness. Nothing happened to him, but he stopped being able to see, without blow of sword or dart. And this blindness continued then forth during the whole of his afterlife. So the Battle of Marathon perhaps is the first example of an acute stressful situation creating a post-traumatic clinical condition. You know who did this one? This is the Guernica by Picasso. So that comes to remind me to say that culturally, stressful conditions, particularly associated with wars, have always attracted the interest of people in cultural terms, in historical terms, because of this facilitation of memory. You always remember better those traumatic events. And of course, we have our own cultural creations that are relevant. So whilst with Bashir is clearly a description of PTSD, even if it is an animation movie. Okay, what is PTSD? How is it defined? So PTSD involves re-experiencing of the traumatic event. I have a cousin who for years would jump whenever there was a bus around, because that reminded him of his experience in the Suez Canal. It involves arousal, avoidance behavior. They don't communicate very well with anyone. People lose the capacity to work. They are very restless, and they have an elevation of those immune proteins that we saw in that mouse. Now, nobody connected this inflammation with their psychological situation. And actually, part of what I'm telling you today was not even published yet. But we looked into the possibility that this is causally related. So in that case, let's think, what do the clinicians do to treat PTSD patients? Which is clearly not a simple question. So the first group of drugs are called selective serotonin reuptake inhibitors. Serotonin is another neurotransmitter. Unlike acetylcholine, it is not degraded by an enzyme, but it's taken back into the nerve cells. And if you block that taking back, you improve the psychological state. The important point is SSRIs penetrate the brain readily because they are lipophilic molecules. They can go through a membrane. Okay. The other family is tricyclic antidepressants. Again, small molecules that are lipophilic. They can penetrate the brain. They do change cholinesterase activity, but this is beyond the point. These are the very well recognized. Now, you'll tell me that does not solve the problem, and you'll be right. Nobody can cure a PTSD patient. A PTSD patient that's stabilized in a disease state, I'm afraid, will remain that way throughout his life or her life. The window of hope is that it takes many weeks for them to get into the state of PTSD. So if you would know initially who is at particular risk, perhaps that can be prevented. The problem is nobody knows. Whenever there is a terror attack, you hear that many people were hospitalized, and of those, 20% or so were just anxiety patients. 10% of the anxiety patients will become PTSD, but no one can distinguish between those and the others. Okay. So these are drugs that are easy to understand. It gets to the brain, it can treat at least symptoms, which is something. But patients who are treated with beta blockers, these are cardiac drugs that are given to control heart rate. They are also improved in their psychological stress symptoms. And this type of drug doesn't go into the brain. Patients who get glucocorticoids, hormones, for other reasons, react again by minimizing their stressful symptoms. So you have drugs that do not penetrate the brain, stay in the circulation, and yet affect the brain. This goes back to the circle that I showed you before. The circulation sends messages to the brain that affects its status. Not only the brain sends messages to the circulation. And now I'm going to tell you a little bit about the research of a student of mine whose picture is on the board, and you may recognize her. So this is what Karen does. And what she's done initially was to ask the question regarding this gene, the acetylcholinesterase gene. What happens if instead of psychological stress, we will create an immune stress? We'll activate the immune system, and then what happens to the cholinergic state? We've just said that a stressful condition changes the cholinergic signal, and that changes the immune system. What if we go backward? So we communicated with friends in Copenhagen who ran a very interesting experiment whereby people volunteered to be injected with this immune protein. IL-6 is the name of that protein, which creates disease-like symptoms. The temperature goes up, you really feel sick. And they took a group of people of 20 years of age and a group of 70 years of age. In Israel, you cannot even apply for such an experiment because the committee would not approve it, but in Denmark they did. And they sent us the blood samples for analysis. And here is what happens. You inject people with this immune protein, and what happens is the cholinergic system responds. So these are the injected people, and their enzyme activity go up in response to the injection. Now, deleting the molecular terms, we are talking about something that starts with the cholinergic signaling, acetylcholine signaling, and anxiety. And we started by saying this changes, in fact, inflammation, immune activity. Now I told you an experiment which does the opposite. Inflammation changes the cholinergic signal. And the cholinergic signal would change inflammation. Inflammation would change cholinergic signal, and so on. Something needs to block all that. And the question we ask next is, what is that something? But before we did that, let me tell you about an interesting correspondence that I had. So I was on sabbatical at Berkeley, and suddenly I get this email from a guy who is a reporter for the ESPN channel, the sports channel. And he tells me about this fellow. His name is Dontrell Willis, and he plays with the Detroit Tigers. He's a pitcher. Reading that, I got off the computer, I went to the lab to ask people, what is exactly a pitcher? I mean, I know nothing about baseball. Okay, so then I learned that a pitcher is a very anxiogenic profession. And several pitchers had to leave their job because it is so stressful. And this guy was the star of the Detroit Tigers. And when I Googled him, I found that he signed up on a contract. He's obviously not a professor. The contract is on $29 million for 3 years. And the contract says that he would be paid 22 of these 29 million, even if he doesn't play. Now this guy came to the manager of the Detroit Tigers and said, I developed an anxiety syndrome, I cannot play. And this breaks the entire group. It's an issue of big news. It interests everyone in Detroit and above that. And this guy says, so I am a reporter. I went to search for a measurement of anxiety. And all I couldn't find is that you developed a blood test, which is a basic research story. And can you tell me if this is commercially available? Because this guy, the Dontrell Willis, was asked by journalists, how do you know that you're anxious? And he said, I took a blood test and they told me that I'm anxious. And the reporter says, I couldn't find that blood test except for your paper. So we started discussing, because I happened to be in California anyway, and he eventually decided to write a big story about stress in competitive sports. So that's a detour for you. But anyway, what we've done was to say, what we've done next was to say, let's suppose that the circulation talks to the brain and vice versa. We know how the gene looks because we cloned it. So we can design a sequence that would be opposite to the sequence of the gene, what we call antisense, and block the gene in the circulation. Would that change things in the brain? Or alternatively, we could treat animals with a small molecule blocker of the cholinesterase. That again will change circulation levels. Would that affect the brain? And what you see here is data in the hippocampus and in the circulation. And what you see here, I hope, is that LPS is an infection model. LPS comes to, it's a shortened name for lipopolysaccharide. Bacteria have a coat which is built of these molecules. So we have this extract of bacteria. If you inject people with this extract, they will get the same infection response that our volunteers did. So here is a control mouse. You inject it just with salt water for control, nothing happens. You inject it with another control molecule and LPS. And now you see a big increase. This is an infection response. But if we block acetylcholinesterase, we don't get it. So then you say, okay, we blocked it in the circulation and we prevented the response in the brain. The same thing happened in the blood. The same thing happens in cells. So now we say, did we invent this? I don't know if I ever told you, but molecular biologists basically mimic what exists already in nature. Except that in this case, we did invent it because we didn't know that a parallel system exists in nature. But since then, we discovered it. So I would like to introduce you to this gene. The orange molecule is a tiny gene. We call these genes microRNAs because they're so small. They were, they were only discovered about 10 years ago, and yet the original discoverer already won the Nobel Prize. And why is that? Because he found a totally different group of genes. Could you tell me what does a gene do? What do you think a gene does? Okay. It produces a protein, right? Okay, no, not right. I mean, this was the initial belief. But here is a family of genes that, unlike what we are used to think, they don't produce anything. We also say a gene is something that activates a pathway, a function. These do not do that. They block the functioning. What they do is they circle around a target gene and prevent it from functioning. They actually induce a destruction of that sequence. So they break all the rules. We also used to think that one gene makes a protein that affects another protein, that affects another protein, a one-to-one connection. These genes work against many targets at the same time. So they really break all the rules. And because we are so interested in the cholinergic signaling system, we looked for possible genes that could block this function, and we found this one. And then we started to ask ourselves, does this gene change immune functions? So let me get back again to the stress response. The vagus nerve sends acetylcholine. So we can react to the stressful insult. Then the body produces too much acetylcholinesterase to quiet things down. This doesn't take forever. So something should block acetylcholinesterase. And this something is this microRNA. Okay? And as you can guess from the cover page of this journal, it does affect the immune function. Okay. But we are here to ask about the brain. So suppose we mimic the function of this microRNA and we block acetylcholinesterase from being expressed. So we'll stop the stress response midway. What will happen? Will that affect cognitive consequences of the stressful response? And these are Galit and Moore who did the following experiment. This is what we call a pattern of a Morris water maze. You've heard that. Okay. So mice that are clever learn to find a platform in a Morris water maze, and then they successfully reach that platform whenever you put them in the bath. Mice that are stressed, even several days later, will be confused, they will run back and forth, they will make what we call retracing errors. They will behave like that engineered mouse in the maze that I showed you. So now what we did was to block acetylcholinesterase. If you do that, the mice remain clever. The stressful consequences of an acute experience depend on acetylcholinesterase. We do that by injecting the brain of these mice with a virus that encodes the blocking sequence. We are just mimicking what nature knows to do. Okay, so this is immediately, right after the stress. What about a delayed stress response? How do you create a delayed stress response in a mouse? And the paradigm there is very simple. All mice get really stressed when they smell a mouse, when they smell a cat. And it's pretty amazing because these are mice that are grown for hundreds of generations in secured animal facilities. And yet, you expose them for 10 minutes to the litter of a cat. They don't see a cat. They just smell the urine of that cat. And within 10 minutes, they become stressed forever, which is quite an amazing process. So what we did was to ask more the same student to bring the litter of the cat she raises at home. We sterilized that sand because otherwise they would kill us at the animal facility, which is a very clean and sterile compartment. And then we injected mice, or didn't inject them, with the DNA blocker of acetylcholinesterase. And then the question is, after 7 days, can we find differences? And we find that in 2 different tests. We test for hyperarousal. Hyperarousal means to put them in a cage, then sound a very strong noise, and the mice jump. And if they're stressed, they jump much higher and much more rapidly and many more times. And if we go on and sound it again, again, and again, eventually they understand that this is nothing. But it takes much longer for a stressed mouse to understand that this is not a real risk. And the other test we did they hear about this one? Okay. This is avoidance behavior. So mice are very curious animals. If you put them in this maze over here, which is called elevated plus maze, they can either be in a sheltered, secure environment in these closed arms, or they can go out and feel really in danger of falling because this is open and unprotected, or stay in the center, which is again an open place. And what we asked was, suppose we injected the DNA agent that blocks acetylcholinesterase. Would the mice show or not show a delayed stress response? Okay. So this was the PhD thesis of Gabi Zimmerman, and this is the elevated maze. And here is the time the mice spent in the open arm. You see that these are control mice, they are not stressed. If you inject them with just salt water, they are terribly stressed. It's like a kid that was taken to the doctor once and got an injection. Whenever you take them again, they really don't like that place. But if we inject them instead with our DNA, then are much less stressed, although they got injected. So the stressful impact was there, but it was less effective. Now we say, okay, now let's attribute a stressful experience, like a noise, like a small electric shock. And what happens is, again, stressed mice are stressed, and if they are injected, they are less stressed. Now let's check how do they dare to spend time in an open field. Again, stressed mice do not, and mice that were injected with our DNA go to the open field easily. This is an immediate stress response, and now, and I needed to tell you that we made a lot of DNAs and only one was effective, okay? And now we check the mice after a week. Consider each ball here to be a mouse. We are checking three things. We are checking their startle response in the sound box. We are checking their anxiety index, which is a calculated achievement in the elevated plasmase. And we are checking their habituation to startle. How long does it take for them to understand that this is nothing? And what you see here, that each mouse is different. Mice are inbred, so genetically they're all absolutely identical. They're like identical twins. But they're raised in a cage and they apparently experience different things. So every mouse reacts differently to a stressful condition. Now we inject them. So not only did we subject them to tests, but we also injected salt water. And the mice are all over the place. You can see that they're stressed. If we inject just a test DNA, that doesn't improve things, which is not surprising. But our DNA blocks the stress response. So again, we say acetylcholinesterase is needed not only for acute and immediate stress response, but also for the delayed response that is reminiscent of PTSD. Now we want to ask, is the immune system relevant to this? That's the question we've started with. So we measure the immune proteins, IL-6, IL-1. These are naive mice. We measure it in their blood. We just take a little blood test. Now we check the stressed mice. They have immune hyperactivation as they go. Now we check stressed mice that were injected with our DNA, and their immune system is fine. So no wonder their brain is quiet, the immune system is quiet. Now we look at their brain. So we look at 3 regions in their brain, and these are control mice, and here are the stressed ones. We stain those nerve cells that are active, and I think you can see that they have many more active nerve cells. But those that were injected with our DNA do not show that. So both in the brain and in the immune system, the question is, who's running the show? Did we affect the brain or did we affect the immune system? And if we did, how did that happen? Okay. So first we said, could it be that we just prevented these mice from understanding that something stressful is happening? In that case, we didn't do anything. All we did is to make them stupid. So we took the brain after 2 hours and measured the activation of nerve cells, and you can see the stressed mice have a lot of activation, whether or not they were injected with the DNA. The initial reaction is not modified. We didn't make them stupid. But after 4 days, or 7 days actually, these are the stressed mice, and those are the ones that were injected with the DNA. So it works in a delayed manner. It prevents the consolidation, that facilitation of traumatic memory that we talked about, not the understanding of the stressful event, which is a very hopeful statement, because that means that you can treat people who are stressed and prevent them from deteriorating into a post-traumatic condition. So the question is, what happens in human beings? We have a collaborator, Alon Friedman, who is an associate professor of neuroscience in, he talked to you, right. So this is an experiment we did with Alon. Very good, okay. So Alon handles treatment of PTSD patients. And we measured, compared the immune protein in their blood to that of controls. And sure enough, the traumatic patients show elevated immune activation. But this doesn't tell us yet that it is associated with their symptoms. So what we did was to divide them to two parts, those with the lower levels, those with the higher levels. And then we asked, let's go to the files, the clinical files, and find the symptom impression of these patients. Is there any association between the immune hyperactivation and the severity of their symptoms? So we looked at re-experiencing the traumatic event. You see that those with high-level immune activity re-experience more. We looked at arousal, same thing. We looked at avoidance, less significant, but the same trend. And finally, we said, let's do an MRI, brain scan, and look at their hippocampus. Now, there is a set of very interesting studies that shows that the hippocampus shrinks in traumatized patients. And actually, this was initiated by the same Robert Sapolsky that I started this lesson by telling you about him. So what he showed was that the hippocampal nerve cells are extremely sensitive to destruction under acute and prolonged stress. Then there were sets of studies that showed that some people get born with a smaller hippocampus, with less neurons, and those would be extremely sensitive to post-traumatic stress, which is a very important study because it tells us a little bit about who is at particular risk. Now, what we did here was to ask, does the volume of the hippocampus relate to the level of the immune activation? And you see that it's highly relevant. So the more immune activation we found, the smaller was the hippocampus of that particular patient. What about the pituitary gland that sends these signals to the circulation? Of course, we saw the opposite effect. So the more aroused they are, the bigger the pituitary gland is. And finally, for control, we measured the volume of the brain cortex. And that, of course, had no change. So this is a selective effect. Okay, so now we say exogenous stressors would affect the brain that would send signals to the adrenal gland, and the adrenal gland controls the production of immune cells. They control the production of immune proteins. Those immune proteins get back to the brain and change its activity. And this is how we get a PTSD pattern with consolidation of the traumatic memories. Okay, now let's ask, how does the immune system do that? So we said that there is an association between inflammation and cholinergic signals. We talked about the body talking to the brain. And let's think about the requirements from a pathway, a chain of command that would give the immune system power over the brain in this manner. So we should say the following. Stressful reactions started because the body detects via some kind of sensor that something is happening, there is danger. For example, you smell fire. That's a sensor system that detects a stressful signal. So anything like that would be required to begin the pathway of reaction. Then the body needs to translate this signal into rapid and robust activation or suppression of inflammation, because inflammation is our readout. And finally, most importantly, this needs to change the way the brain acts. So I'd like to tell you about a system that we all carry and which fulfills these functions. It is called the toll-like receptor. Toll-like because they shaped like a bell. So for who the bell tolled. The toll-like receptors were discovered in flies. And people started studying them, and eventually, now we know they represent a big and very important family of biosensors. We have a toll-like receptor to detect pressure, and another one to detect infection by viruses, and a third one to detect infection by bacteria, or damage or death of cells in the system. You name it, we have a toll-like receptor for that. And this is how they work. Like, suppose something bad happened, cells in the body started to die. There was an injury. Then DNA is released. That DNA can bind to a receptor, get into the cells, and activate a toll-like receptor. Same happens with viruses. We get infected with a virus, there is a toll-like receptor that would activate a lot of response actions. So this is a sensitive monitoring system. It induces rapid and robust activation. Okay? And it can either promote or suppress inflammation. So we said we have our culprit. That's what we think happens. But still, we need to prove it. So how do you prove if the toll-like receptor sends messages to the brain? Toll-like receptors are only in the body. We said, let's assume that our DNA affects the toll-like receptors. We made a prediction. If that is the case, then it would change acetylcholinesterase, this is our target, in the circulation when we inject animals with it, and it does. But it would not be able to do that if we block the toll-like receptor, if this functions through the toll-like receptor. So we co-inject the animals with our DNA and a DNA that was known already to block the toll-like receptor, and then we don't see any effect. So now we say, aha, we block the acetylcholinesterase, but that effect is controlled by the biosensor system called toll-like receptors. Suppose this is the case, then how can we use it to our benefit? And now, I apologize, but only now do we get to the autoimmune diseases. Okay. I told Karen that I'll teach you about autoimmune diseases, and the background took a whole hour. So this guy on the screen now, his name was Thomas Willis, and he was the king's physician in England. He was a very wise man. He got this important position after he discovered why people in the villages become crazy when they prepare cider. So every summer they would pick the apples and ferment them and put them in lead-covered barrels, and some of those people would get totally crazy. And it was only men, because the women were not involved with cider preparation. And it was a particular breed of men, so it was clear to him that this was genetic. And apparently, these are people who carry mutations that make them very lead sensitive. When he discovered that, and they stopped, and they didn't let them drink the cider anymore. That solved the problem right there. And he got this wonderful position of the king's physician. And he wrote a story, he wrote a paper, a scientific paper, which a friend in England translated for me from Latin, because in the 17th century you wouldn't write scientific articles in English. It was always in Latin. And there was no space limitation either, because he wrote a very long and tedious story about this little lady who he interviewed, and she gets up in the morning and she can function, she can walk, she can talk, she can do the household work, which is what she was born to do. That's, the paper says that, so now we know. And by noontime, she's so tired that she cannot walk. She becomes as dumb as a fish, and she's terribly fatigued. So what he described was a disease that today we call myasthenia gravis. And the characteristic feature of that is progressive fatigue. Myasthenia gravis is known to everyone from the Walt Disney Droopy. And Droopy was drawn by Walt Disney because he had a friend with drooped eyelids. The billionaire Onassis, some of you might remember his pictures with dark eyeglasses. He wore eyeglasses because he suffered the same disease. And he died because of it. He got into acute crisis on a ship, and that's why he died. So what is myasthenia gravis? Think about nerve cells sending a neurotransmitter, acetylcholine, to the muscle over here. The muscle has receptors for acetylcholine. Otherwise, we wouldn't activate the muscles thanks to this chemical. And the muscle also produces acetylcholinesterase, and this protein degrades acetylcholine. And it's always a balance. Now suppose that the body blocked some of these receptors by erroneously recognizing them as non-self. So the body produces an immune response against its own protein. This is the essence of an autoimmune disease. What happens to these people is they don't have enough acetylcholine being sent. And therefore, the whole system gets out of balance. Too much acetylcholinesterase is being produced. Their muscles get weaker and weaker. So what is done is they get treated with small molecule inhibitors of acetylcholinesterase. And when they take a pill, then acetylcholine levels go sky high, and they feel very well for a couple of hours. But the body translates that as an acute stress condition. So there is overproduction of even more acetylcholinesterase, which creates a vicious cycle. And we said, we clone the gene. Instead of putting a chemical in, why don't we put our DNA? We'll block the gene, which is a much more natural event, and we'll need many less molecules because we're not blocking the protein, which is the bottom of a pyramid, tens of thousands of molecules. But we only need to block the RNA, which is hundreds less in terms of number. So we injected sick rats, autoimmune rats, with our DNA, and these are the black dots. And what you see here is that the rats developed excellent muscle tension. They could walk. We walked them on a treadmill. They gained weight. They really improved. If we injected those rats with the clinically recommended dose of the small molecule that those patients receive, then they would be improved for a couple of hours and deteriorate, just like this happens to patients. Patients have to take about 10 pills a day because of this response. Okay, so then we say, that's a good question. At least 24 hours, in some patients up to 72 hours. We'll get to that. So here is an example of a patient. We got into clinical trial. And in the last half hour of today, I want to tell you a little bit about clinical trials, because I heard that you heard a little last week, and I'd like to deepen that. So this guy suffers from myasthenia gravis. He's a patient at Hadassah Hospital. And you can see that he has difficulties to open his eyes. takes 10 pills a day. And apparently, this doesn't help. So he volunteered into our clinical trial. Instead of these drugs, he only gets our experimental drug. And this is how he looks. I mean, people compete on the right to participate in these clinical trials. The dose is much, much lower. Even if you calculate the molar amounts and how many molecules, it's like 1,000-fold lower. 1 month after the test, he still looks much better. And we still need to prove if that reflects a change in his immune system, which is an interesting question. And here is another guy. So this guy is from Manchester. Can you see it? Okay. Right. So this guy is suffering from a problem, a weakening of the swallowing muscles. And the test is he needs to drink a glass of water. See here, this is our drug, and it takes him 9 seconds to finish the glass of water. This is the traditional drug. Note the count of seconds. He needs 19 seconds to drink the same glass of water. 28 hours after this treatment, he functions as if he's under treatment. And if he gets no drug, he needs over 30 seconds to drink half a glass of water because he needs to stop and swallow every time. His muscles are really weak. Okay. So, the system works over time. In such a case, you need to run faster. And which is the system that works over time? We call it the innate immunity system. It is not an acquired immune response. It is not that these people were exposed to anything. The immune system got a wrong message, and it works over time continuously, just like the Red Queen from Alice in Wonderland. So you really need to run twice to get to a certain place. Do we know about other autoimmune diseases that affect people who are aged? And this guy, his name was Henrik Sjögren. I will learn how to spell that correctly. And he's a Swede, and he identified a disease that is called Sjögren disease until today. And the disease is affecting mainly women, like 90%, and mainly people above the age of 50. And what it does is it stops salivary function. People do not have tear fluids, and they suffer dryness of the mouth. And it's not a lethal disease, but it's very, very uncomfortable. People feel bad about it. And this is exactly like myasthenia. It's an autoimmune disease where the antibodies in this case are produced against the acetylcholine receptor in the salivary glands. And then we said, okay, in that case, our DNA should be very helpful in that case as well. So this is what happens to the eyes of Sjögren patients. And I can shorten the long story and tell you that this is exactly what happens. We can take mice with Sjögren disease and improve their salivary function beautifully. There are other autoimmune diseases that affect the same system, like intestinal bowel disease. Our intestinal system is innervated by the vagus. An imbalance in that system would create either an autoimmune or just hyperactivation of the innate immune system, which is, again, a non-lethal, most cases, but very long and tedious and very unpleasant disease. And last but not least, in the list of diseases, I'm sorry I'm talking so much about diseases. But again, this is the title of the course, so maybe I shouldn't be sorry. What happens after stroke? So this is a physical impairment, which again affects the brain, but also the body. And it creates a very massive immune reaction, inflammatory reaction. But when you look at the patients by their immune markers, like here, every circle is a patient, and these are the patients, here are controls. You see that about 20% of the controls have hyperactive immune system too. So this cannot serve to discriminate between stroke patients and controls. If we measure the cholinesterase activity, which was the PhD of Shanish and Har over here, then we get a much better discrimination. So this system, again, is relevant. So what happens to cholinesterases in stroke patients? You have a distribution of activities, which is the case in any biological parameter. In control volunteers of the same age, the same weight, the same symptoms other than the stroke, you get most of them at this level of activity and others with some a little bit less, a little bit more. These are the stroke patients. You see immediately that here the balance got shifted, but because of another reason. It's not hyperproduction of acetylcholine, it's too little acetylcholinesterase. But the consequence is the same. Too little acetylcholinesterase will create a risk to not survive the year after the stroke. Why? Because acetylcholine, for example, innervate the heart, and these people may get a heart attack and die. So then you say, what happens to people who have a lot of acetylcholinesterase? Just like controls, although they are post-stroke. In this case, they will suffer poor recovery in terms of cognition. So after a year, they become a little bit like Alzheimer's patients because they have too little acetylcholine. So we need to bring them from here to here to lose the risk but not create a new risk. How do you do that? What do you think one should do when you have too little of an important protein in a patient? What do you suggest? What would you do? Ken?
Well, the protein metabolizes the
Mm-hmm.
original hormone. So would there be a way of repressing the production of the original hormone so that the proteins that are produced
That's an interesting suggestion. Thank you. So one way would be to block the production of acetylcholine, but we just said that we need that. So that would be a problem again. So what would be the other option? Continue the same line of thought. If you cannot block the production of the neurotransmitter, what can you do?
maybe some of the acetylcholine be already blocked and you could
You could add the protein that is missing. Okay? So this is done in some cases. Insulin is a protein. When it's missing, we inject it. People with Gaucher disease suffer, another protein is missing. We inject it. So I'm happy to tell you that this protein is produced in a company in Carmiel called Protalix. They have conditions for protein production under very strict conditions, approval for drugs, and they do that in plant cell culture. So the cultures are from carrot. You can see from the color. And they hang them on metal hangers. The whole factory is like two size of this class, and they produce 10,000 liters of this culture, and they can purify the human protein from a carrot cell culture, which is a major advantage because you don't even need an incubator. You don't need to elevate temperature. Plant cells grow at room temperature. So we are discussing with this company. We sent them the DNA because we cloned the gene. So they signed a license with the technology transfer company of the university. They are producing the protein, and now we are discussing how to treat stroke patients so that we'll bring them above the acute lethal risk without causing long-term damages. So that brings me to the issue of how long do we produce drugs under a structured set of regulations? And I looked because Karen told me that Steve Wilton talked to you a little bit about that. So I made a little search, and Karen helped me, and then I made another search. Actually, I was quite surprised myself. So it's only about 100 years that any drug needs a permit to be produced, to be sold, to be administered. Isn't it surprising? For people who are involved with that, it is surprising. So for you, I mean, we sort of think that this has been the case always. You want to produce a drug, get a permit. You want to sell a drug, get a permit. You want to administer, test that it's okay. No. So there were stories that were horrible. People were selling drugs just by talking to others. And for example, a famous story that I heard is a place in the United States where there was a radioactive spring. So the water was radioactive. Anyone who drank that water would get over any bacterial infection, intestinal infection, beautifully because the radioactivity kills the bacteria. But if you take it too long, you'll also kill your own bones. And this was a very popular drug. People were running around selling this poisonous water, and people were taking it. So these are examples of the drugs that were sold until 1906. I'm sure that snake oil is something really helpful, but I doubt if there was, at least in the United States, the 4,000 years of trial and error that established the Chinese medications. So what happened? Then the United States of America decided to create a national laboratory. And this national laboratory checked reagents first for purity. Chemistry already existed. They could take something and ask if it's contaminated, and it was. So that was the very first function. But to cut a long story short, because my son is getting his graduation today at 5 o'clock in Givat Ram. Yeah, so we... Okay, do you want to go with us? We can offer a ride. Okay. So this is what the FDA expects today from anyone who wants to develop a drug. So first you do preclinical research. What I told you about those rats that had an autoimmune disease is a preclinical research. Or when you look retrospectively at samples of patients, like the stroke patients, like PTSD patients, this is also a preclinical research. It does not need an FDA approval. And then you get into clinical studies, and that would be phase 1, 2, 3, and 4, and even after that, there's a lot of checks and balances and regulations to make sure that the drug is safe for use. So what are the goals of each phase? Preclinical trials would be mainly in animals or tissue culture. What I told you about is classical. Can anyone estimate how much of the expense of the production of a new drug goes into preclinical research? Percentage. I'm sorry? 2%. I was shocked when I heard that too. Why? Because they'll tell you, the drug companies will tell you, to develop a new drug costs $1 billion. Why? It doesn't really cost $1 billion, but only one out of that many succeeds. And the cost is for all those that failed along the way. And when you complete your trials, then only then you start selling it. And the salesman is where all the money goes. So this was very disappointing for me, but it's true. First, preclinical research, then you have to find a partner. A partner is a company that would like to handle the clinical studies. We are basic researchers. We don't do clinical experiments.
Can I ask a very small question?
Sure.
This has a little bit to do with ethical issues, but don't sometimes, just some of the time, pharmaceutical companies donate some monies for these clinical trials, and then it's a little bit like unethical or whatever?
It is watched over very carefully in the Western society, less so in East Europe, even less so in Africa. Did you see The Conscious Gardener? There's an excellent movie exactly on that issue by John Le Carré. Yeah, highly recommended.
Okay.
Talks about how people in Africa would get their AIDS medicine only if they volunteer to be tested for another drug totally.
Okay.
So, which is totally unethical. We'll get a little bit into that. Okay. Phase 1 clinical trial is to check in a small group of healthy volunteers just to make sure that the drug is not dangerous. So this is a safety test. Phase 2 will be larger, and you would treat the patients. Phase 3 is still larger with a broad trial, with a large sick population, and how many is an issue of discussion with the FDA. So the Federal Drug Agency discusses now with anyone who wants to develop a drug and directs them and gives consultants and advice, and if they would not accept your conclusion, then they would delay the decision. After these 3 phases, you have phase 4, which is a retrospective look. So the drug was already approved. It was released. You need to make sure that it didn't cause any damage to those patients who were treated with it. Now, the situation got to be this way gradually. In 1938, it was decided that new drugs should be proven to be safe before selling them. And the government would control. They would not allow to sell them otherwise. It covers also cosmetics and therapeutic devices. Cosmetics is raising a lot of anger worldwide because of the use of animals. So that may be changed. The toxicity information must be given. If you buy any therapeutic agent, you'll find the risks and the side effects listed. And you need a prescription. I cannot buy a drug just like that. And you need to show that this is honest and convincing. So initially, 1906, that was when the FDA was created. 1953 only in 1953, they started checking in factories that the production is okay. And manufacturers like Teva must produce information about the analysis of the drugs they produce. The 38 we discussed. 62 is a timeline. In 62, thalidomide was found after it was approved to cause terrible birth defects. It was approved for headaches and depression, but there was no prevention from pregnant women to take it. And that was, sorry, okay. So that happened in other countries, not in the United States, because the United States did not approve the use of thalidomide for the good reasons that they said there was not enough information. And they saved their population from this horrible thing. And when that was known, the Congress said you need to watch over it even more carefully than you did before. Okay, so sulfa. When I was a kid, this was the only antibiotic that was available. And sulfa was given to any disease. And the law at that time did not say you have to prove safety. They dissolved sulfa in diethylene glycol, which is by itself poisonous. About 100 people died, mostly children, and then they redid the regulations. This is the act, and we will skip that. And I'd like to tell you about this guy. Frederick Banting was an MD who didn't want to take care of patients. He had a passion to solve the problem of diabetes. And he cut off the pancreas from dogs and injected them with extracts of pancreas from other dogs, and that's how insulin had been discovered. But he could not produce insulin reproducibly at the same quality. And then Eli Lilly, the very big drug company, got into the picture and started selling insulin. At the same time, penicillin was discovered and started to be sold. Again, penicillin has to be purified, and it was after damages to many patients when that was discovered. Okay, other drugs also showed problems, and each of these problems brought more budget, more manpower to the FDA, which became a very big body with very powerful effect in the States. Now, just a little bit about thalidomide. It was approved in Europe and Canada for impairments in sleep and nausea. And nausea, these are pregnant women. The discoverer got a medal for this important drug, which is actually a very good drug, as long as you don't give it to pregnant women. And the FDA discussed it and decided that the safety data was not sufficient, and they didn't approve it. So this is a very strong example that they keep telling you about. In '62, they added antibiotics as a drug. And one very important issue. Well, how does the public influence the FDA? And a wonderful example is that of AIDS. The AIDS epidemic was initially a lethal disease. And the public demanded that research would be supported and drugs developed. And it worked. So the NIH, the National Institute of Health, supported a lot of AIDS research. The proteins involved were purified, crystallized, small molecule inhibitors were developed to block the viral proteins. And today, AIDS in the Western countries is still a very strong disease. It's important morbidity. Patients have to take a lot of medication, but it's not lethal anymore. It's a chronic disease. So this is a marvelous example. The example of Alzheimer's disease is not there yet, because the public equally demanded cure for Alzheimer's, and the problems there we discussed in another lesson. It's very difficult when you diagnose it so late that most of the nerve cells are dead. Orphan drugs. Orphan drugs are drugs that are treating less than 1 million patients worldwide. Myasthenia gravis is less than million, so that's why it is an orphan disease. Anti-tampering act means that you really have to be honest in developing the drugs. And in '87, they were further expanded because molecular biology came up. And now we have drugs that are proteins, which we just discussed. These are called biologics. So that's a different department in the FDA. Okay, so I think that we are going to stop here. I'll put all of this presentation in the website so that anyone who wants to know more can read it. And I think that this covers my part in this course. You have 2 other guest lecturers in the next 2 weeks. You've been a wonderful class. It's the first time in my life that I am teaching out of the Faculty of Science, and it's a very positive experience. So thank you.